Greetings, I'm Mark Brenner. I'm a professor in geological sciences at the University of Florida. And as always, it's a great pleasure to be here at the Archbold Biological Station, in particular visiting Lake Annie. First time I heard of Lake Annie was in 1975, and that was the first year I started as a graduate student at the University of Florida, and it was the year that Bill Watts published a paper on Lake Annie. He had taken a very, very long sediment core in the lake and studied the pollen in that sediment core, and it went back into the late Pleistocene, that is to the last ice age. And he was able, essentially it was the first good glimpse we got of what ice age ecology in this part of Florida looked like. It's a really spectacular piece of work, and subsequently he went on, along with colleagues, to do work at several other lakes. So we're on the Lake Wales Ridge here. We're in south central Florida. We're sort of at the terminus of the Lake Wales Ridge, up in a relatively high point, surrounded by scrub, as I say. And some of the lakes here are just spectacular in that many of them are clear water, low pH systems, relatively unproductive, just ideal places for recreation, but also wonderful wildlife areas too. And my personal interest is in paleoecology, paleoenvironments, and one of the things that we'd like to make clear is that the work that we do in studying the past conditions in these lakes has a lot of application for managers in terms of managing lakes and managing local terrestrial environments too. One of the things about Lake Annie that makes it really special is it is quite deep for a Florida lake. It's about 67 feet deep. It's a 90-acre lake. And one of the ironies about working in Florida lakes is that the smaller sinkhole lakes are fairly deep, and a lot of our huge lakes like Okeechobee and Istokpoga are relatively shallow. The interest in the deep lakes, of course, is that because they're deep, they've held water continuously back into the last ice age, whereas many of the shallow lakes first filled with water probably about 8,000 years ago in most cases. Deep lakes that are situated on the Lake Wales Ridge and the Trail Ridge farther north in Florida have been really informative about long-term changes in the landscape here. For instance, if you were to find yourself here during the end of the last ice age, you would probably have found it would have been much cooler and probably much drier. Ceratiola, or rosemary, seems to have been much more prominent on this landscape, whereas today it's confined to these upland, very, very well-drained areas. And then the other thing that we see is as you come into the Holocene, which began a little over 11,000 years ago, we see a lot of oak on the landscape, and then about halfway through the Holocene, maybe 5,000 years ago, we see that pine maybe begins to outcompete the oak. Pine becomes more prominent on the landscape. But there are lots of other kinds of questions that can be addressed. And one of the things that I think is really interesting, when I was a little kid, we always saw Smokey the Bear commercials, and we were told, stop forest fires at all costs, stop forest fires. Fires are bad. And what we're finding by going way back in time before humans were a major presence on the landscape is that fires have always been an important part of some of the ecology here. And so you can do a number of things, like you can bore into trees and look at tree rings and scars, fire scars on trees, but you can also look at charcoal in lake sediment cores and see, in fact, that fires have always been part of the ecology. And I suspect, in fact, that people who are doing controlled burns now, prescribed burns, are using that information to explain and justify why they are doing these periodic burns to maintain the ecology of places like the Archbold Station. So oftentimes when we go to the field to collect the sediment core, often we'll do some work ahead of time to determine some, what I would call ideal spots in which we'll take a core. Here at Lake Annie, we had the good luck to know in advance because we had taken cores here before. And so what we did is we went directly out to the buoy where data are being recorded all the time from the lake itself, and we decided it'd be very nice to take a core right at that spot so we can link, again, the modern data with the sediment data. So the thing to do is we take the boat out to the site, We anchor very well. In this case, we were able to tie off to cables that are coming off the buoy. And then the next step would be to put together our coring. One of the first things we do is we have a little sonar device and we get an accurate measure of the water depth, because this will tell us where we need to start our drive and capture that topmost meter or meter and a half of sediment. Once we have that established, we can do those calculations, and then we begin to assemble our coring apparatus, which consists of a polycarbonate core tube that's about 2 meters long, a series of coring rods, 1.5 meters long, a couple that are 1 meter long, and a piston and basically a cable. So what we do is we put the piston in the bottom of the tube, connected to the cable. We put a head on that tube, connect it to a rod, fill this thing with water, and start going down, passing the cable and the rods down until we're at about 30 centimeters above the sediment water interface, above that sediment surface. Then we'll tie the cable off and prepare to go ahead and collect our sediment. So after we collect cores, particularly these uppermost sediments that are not very well consolidated, we bring them into the boat, seal them up, and we try to keep them in a vertical position so that the sediments don't slosh around. What we'll do then is we'll cart them back to shore, always maintaining them in that vertical position. We get our cores back to an area where we can extrude them. We mount them in a tripod, and this enables us to extrude them vertically by pushing upward on the bottom on the extruder piston in a very controlled way. And we collect the material that we're extruding in a tray that's at the top of the core tube. And then we collect that material into labeled Whirlback bags or cups that are labeled, so we can transport that material back to the laboratory. And what's kind of fun and interesting about this kind of work is that we can't buy a lot of this kind of equipment off the shelf. In fact, almost everything that you've seen being used today was designed and built by Jason Curtis, the fellow you saw coring with me out in the boat today on Lake Annie. We are interested in a number of things in Florida lakes. We've done a lot of work for the water management districts. Much of that work was focused on looking at trophic state changes, particularly in shallower lakes. That is to say, we're looking at how nutrient enrichment in those lakes has changed, largely as a function of human disturbance, septic tanks, sewage disposal, fertilizers, and so forth. And what we could do is with relatively short cores, we can go down through the sediments there, we can make measurements, geochemical measurements, measure things like phosphorus, nitrogen, and carbon directly. We can date those cores using lead-210, and then we can work with people who are diatom specialists, and the diatoms are algae that are preserved. They have silica shells. People can identify them to species level, and they can be used as indicators of changing trophic state in the past. So the benefit of that is that we can give managers some idea of what the baseline conditions in these lakes were, say, before the turn of the last century, before 1900, and let them know how much the lakes have changed, and possibly even tell them whether there might be any chance of restoring them to a previous state. So oftentimes when we do a project, say for one of the water management districts, they have a very specific goal in mind, and we'll say, we're gonna go out and grab a core or multiple cores to look at diatoms, because we can use the diatoms to make inferences about past water quality. But we also realize that we and other colleagues have other interests in those cores. Some people might want to look at stable isotopes in the sediments, or things like thecomebians or cladocerans. So we always keep extra material well labeled, well archived, and refrigerated, so that down the road people can work on these same cores that are often dated with lead-210 and radiocarbon. We've had occasion to have people approach us and ask for material from some of our lakes where we cored 30 years ago. And in fact, they are today using technologies that didn't even exist at the time we took our cores. So that tells you, in fact, how important it is to keep these things archived and preserved so that others can use them. I think one of the new, exciting, what I might call, oh wow, things that's coming down the pike in conservation, paleobiology, is people are starting to look at environmental DNA in aquatic systems, in the water column, but also in the sediments. And so instead of having to go out to the lake with a plankton net every month, or take water samples and look under the microscope at different algae, what you can do is you can take a sample, extract the DNA, and in one fell swoop, get a picture of everybody who's been there. And so the applications might be something like if you have an invasive plant in a lake or something you consider to be an invasive fish, you can test that hypothesis because you can take a core, you can date that core using lead-210 and radiocarbon, go back in time, and see if you find the DNA of that specific plant or fish back in time. If not, if it's something that only appears in the very recent, you can be pretty sure that it was introduced recently by humans and in fact is an introduction that is a pest species in your system. So I've really had the good fortune of working with many people, international colleagues, people throughout the US from different institutions, many students, graduate students, undergraduate students who've been helpful in the field or people who are focusing on their dissertations and theses. And we've addressed a number of what I consider to be very interesting questions. So for instance, in Southwest China, one of the things we looked at was the development of the Asian monsoon. And then later in the Holocene, we were also focused on how humans had really transformed the landscapes, changed the lakes, caused a lot of erosion in those watersheds. Elsewhere, for instance, in Madagascar, we did a project there that's ongoing, looking at when the island was in fact first colonized. We're trying to get a handle on when people first came onto that island because it was relatively late. It looks as if it was relatively late in the Holocene. And then where I started out was in the Maya area, in the lowlands of Guatemala, and subsequently I worked in the Mexican part of the Yucatan Peninsula. But I began my work there in the mid-70s at a time when people were really becoming concerned about human impacts on the environment. 1970, I believe, was the first Earth Day. And so when I was starting, what I was trying to look at was how the ancient Maya had deforested the landscape, caused massive soil erosion and sedimentation in the lakes. And then about 10 to 15 years later, as climate change was starting to rear its ugly head, we got involved in looking at past climate in the Holocene, particularly changes in rainfall amount in a number of areas, in Haiti, on the island of Hispaniola, in the Yucatan Peninsula, in Mexico, in Guatemala, and also in Venezuela. And one of the very interesting things that we discovered there was that the classic Maya collapse in the 9th century AD appears to have been associated with some very profound and protracted droughts. And so our interests have been in what I would say are these relationships among humans, climate, and environment. And we gain these insights that I think will be very helpful in managing going forward.